Hello everyone. Today's topic of my tutorial is protein 3D structure modeling by using X-ray crystallography. In the previous tutorial, I already explained about the homology modeling, that is one of the in silico technique which is also used to predict the 3D structure of the protein. Here in this tutorial, today we will see about the X-ray crystallography. So here we see the more three important points. What is the need of X-ray crystallography? What does it mean? And how it would be performed. So basically the X-ray crystallography, it includes the technique name itself says that it uses the X-ray, it uses the crystal. So first it is needed to understand what is X-ray, what are the advantages of X-ray. X-ray, it is used to identify the 3D structure of protein, and why the crystals is necessary to use to identify the 3D structure of protein. So first we learn about what is X-ray, why X-ray is used, and what are the advantages of the X-ray being used as a source of radiation to identify the 3D structure of protein. So the main advantage, first main advantage of X-ray is that it is an invisible light. It has the high penetrating power. It is an electromagnetic rays. So the another advantage is also that the this X-ray is illuminated to the protein crystals. So why only X-ray, not other rays like alpha ray, beta ray, or any visible rays could be used to identify the 3D structure? Because the wavelength of X-ray is very low. That is, it ranges between the zero point 0.1 to zero point 0.2 nanometer, and the distance between the two atoms present in the protein crystal, it is very likely much that of the wavelength of the X-rays. That is, it is range between the zero point 0.1 to zero point 0.3 nanometer. So that's why the X-ray is used to identify the 3D structure of the protein. So next word is the crystallography. In this word there are two subwords. First one is the crystal and another one is the graphy. So graphy means the you directly can't get the molecular or atomic level structure of the protein. First you get the some graphical form of the graphical form from which is recorded on the detector, and thereafter that graphical information you get converted into the atomic structure. That's why it is called as a graphic. So another important, most important terminology in it is the crystal. So crystal is used to identify the protein 3D structure. That crystal is made up of the protein and some other chemicals which is used to make it crystalline. We'll explain in the later slides. So why there is necessity to make the crystal? Because the all the biomolecules which are present in the cell, they are not present in the single conformation, single orientation. They change their bonding, they change their conformation. There may be some due to their interaction with another protein, they change their in conformation. So if you make that protein in a fixed conformation, then it is more easy and more accurate to identify the 3D structure of protein. So to make that protein molecule in one state, in one freeze state, it is necessary to make it in crystalline form. So that crystallography is basically used to identify the 3D structure of the protein. And why there is a necessity to identify the 3D structure of the protein? Because the 3D structure of the protein is the functional protein. It helps in the identification of function of the protein. It helps in the identification of whatever the what are the alpha helix and how many beta sheets, how many alpha helix are present in that particular targeted protein. So if the X-ray crystallography, it would be defined as it was a method. It is one of the proteomics technique is used to determine the arrangement of atom within the protein crystals. So how it would be identified, we'll see in the next slides. So next comes to the most important point of that. It is what is the principle of the X-ray crystallography. The principle of X-ray crystallography is based on the two important phenomenon. or the two important points you should keep in the mind. That is, first one word is the diffraction pattern, and another one important word is the intensities of the spot. Here you can see that two diagrams which explain the principle of the X-ray crystallography. So here in the first diagram, suppose, consider that or imagine that this is a protein crystal, and this protein crystal contains this number of atoms which is placed in scattered throughout that crystal. So when this protein crystal is exposed to the X-rays and it struck on the particular atoms present in that crystal, protein crystal, it will get refracted. It will get refracted. It will change their angle of reflection, and this is nothing but called as a diffraction pattern. Okay, so suppose this is a ray. This is a ray coming from the X-ray source. It come like this. It come in this direction. It struck on this. It is not going straight forward by throughout this atom, but it will struck. It will hit on that. It will hit on that atom, and it will get change their direction, and that will be recorded on the detector which is present next to the crystal. So this is the first important phenomenon, first important point which would like to keep in mind when you write the principle of X-ray crystallography. So another important thing I was tell that the intensities of the diffracted beam of light. So whatever the rays coming from this atom will get struck or will get bombarded on the detector which is placed next to the crystal to identify the densities of atom present in that particular crystal. So this concept is clear in this second diagram, that here in this second figure you can see that there are some small spots, but there is a variation in the size and the intensities of the spot. Some of these spots are very dense and larger size. Some of these spots are very lighter and very small in size. So how it would be obtained? It has the correlation with the density of electrons present in the orbital shell of the atom. So whatever the atoms present in the protein crystals in its orbit, it carry the electrons. So if there is a higher density of electron present in the orbital shells of an atom, then you will get the more intense, more darker band. And if there is a less electron in the orbital shell of the atom present in the protein crystals, then you will get the smaller and lighter band. So basically the principle is governed by these two phenomenon, that is, one is the diffraction pattern, and from whatever the rays coming from the X-ray source, it gets struck on the atom, it will get refracted, and whatever the angle is changed by that particular incidence ray, it is recorded, and that is used to identify the protein structure. And second one important point kept in mind is that the intensities of the diffracted rays coming from the atom in the protein crystals. So you must write these two important points in the principle. Next, we'll see about the mechanism of X-ray crystallography. So, basically, the X-ray crystallographer, it is one of the instrument which is used to identify, which is used to predict the 3D structure of protein. So, it contains the source of X-ray. Then it contains next to source of X-ray, it contains some filters, so that whatever the rays coming from this X-ray source, it will get filtered, and only the X-ray will pass towards this crystal, which is placed on the gynometer as a supporting material, as a supporting source. So the X-ray is coming from the X-ray source, it will get hit on this protein crystal, and the data will be recorded on this detector screen or detector plate. So the X-ray source basically used in the crystallographer is the X-ray tube. after the X-ray source, there is a some filters are placed next to X-ray source that is made of generally the lead filters. So what is the function of that lead filters? It filter the radiations coming from the X-ray source, because there may be a chance with the X-ray, there may be a chance to transfer to some other rays like visible rays towards the protein crystal. So to avoid this, here the filter is placed and that can function as a that can filter the X-rays. Another important function of that lead filter is that, suppose for example this is the source, and from this source at a time, in a single time, number of X-ray beams, number of incidence X-ray beams will come out. But to find out the more accurate structure of the protein, it is necessary to pass a single beam of light in a single time. So the function, so this function could be performed by this lead filter, which is placed next to the X-ray source. So here we will more understand about how the protein structure will be predicted more accurately. Suppose this is a protein crystal. This is a protein crystal. So when it is illuminated by the X-rays, it is not completely in a single time. It is not completely exposed to the X-rays. It is converted, or it is broken down into a small imaginary cells. Means it is not physically broken down into small cells. It will get the destruction in the protein, and there is no meaning to identify the protein structure. But it is imaginarily broken down into a small cells, that is called as a cell. Suppose in this diagram you can see that this is a total protein crystals, and it is divided into a small cells. This is one cell, this is another cell, this is another cell. So when it is illuminated by the X-ray in a single time, the X-ray will only focus or will only detect what are the atoms present in particular this cell, so that it could be more accurate to analyze the protein structure. In the second round of analysis, this part of the crystal will be illuminated to the X-ray beam. So like this, whole protein crystal will expose to the X-ray beam. So it also can move in all direction, because suppose this is a protein crystal and the X-ray is coming out from this direction. It only not because in that crystal the protein are may present at this side or this side. So this crystal will rotate slightly like this, so that each and every side, every edge of that protein crystal will exposed by the X-ray beam. So this is all about the mechanism of X-ray crystallography. Next we'll see the how it would be performed. So the first step in while performing the X-ray crystallography is the isolation of pure form of protein, because when you isolate something from the live biological material or the cell, it has so many compounds. It has the DNA, it has the RNA, lipids, fats, and all these biomolecules and metabolites are present in cell. So we need only a pure form of protein, because purity is more important point while you are determining the 3D structure of the protein. If there is a contamination of something, DNA or the RNA in the protein sample, then there will be some disturbances while identifying the fine model structure of the protein. So first important point while isolating the purity and performing the X-ray crystallography is that the purity of the isolated protein would be vary between the 90 to 95%. It should be 90 to 95%. Less than 90%, you can't use it for the X-ray crystallography analysis. or to identify the 3D model. You have to perform this step again and again to get the pure protein, pure targeted protein. Next one important thing is that the pH and the temperature. When you are isolating the particular protein from the cell, the pH and the temperature after isolation and during isolation, it matters because it requires some fixed pH range or some fixed pH value and temperature so that it is stable at particular pH and temperature. If there is a change, certain fluctuation in the pH and temperature, the protein will get denatured, it will get deactivated, it will change their conformation, or any kind of conformation changes may take place in the protein due to fluctuation in the pH and temperature. So that the purity, pH, and the temperature, these three are the most important thing when you are identifying the protein structure using X-ray crystallography. And the next step is the crystallization. And crystallization is performed using the various chemicals like PEG, that is polyethylene glycol, various salt solution, and various organic solvents which is easily available in market. It is used to aggregate the protein, to crystallize the protein. So the first important thing is you must to isolate the pure form of protein, stable form of protein, and another important step and the second important step is the crystallization using various chemicals like PEG, salts, and various organic solvents. So third step is, before going to third step, the important step is the crystal cryoprotection. This is an important point because it, why there is necessity to do the crystal cryoprotection by using certain chemicals like glycerol, ethylene glycol, or MPD, like means methylene pentadiol, because when the crystal is exposed by the X-ray beam, the X-rays are coming towards the crystal, it will get heated. Continuously it is exposed to the X-rays, so it will get heated, and to avoid the overheating of that crystal, the liquid nitrogen flow is used to cool down that crystal. And the overcooling also hazardous, and overheating also affect the protein structure present in that crystal. So to avoid the overheating, we use the certain cryoprotectant, and to avoid the overcooling, we treat that crystal using various cryoprotectant agents like glycerol, ethylene glycol, etc. And the next important step is, once you prepare that crystal, it may get exposed to the X-ray beam. The X-ray is incident from the X-ray source. It is first the X-ray is get filtered, and then it pass through the once it pass through the crystal, it is get struck or hit on the X-ray crystal. This X-ray crystal cells, and from that X-ray crystal it get diffracted, and the data will record on the detector plate. So here is important thing that when you are exposing the crystal to the X-rays and recording a data on the detector plate, the distance between the crystal and the detector should be approximately 150 nanometer, or should be less than 150 nanometer. Suppose this is a crystal and this is a detector. The distance between the detector and the crystal, it should be 150 nanometer, because this is according to the Bragg's law. All of you, I think it is known to all of you that according to Bragg's law, the distance between these two parts of the crystallographer, it should be less. If the distance between the crystal and the detector is more, whatever the rays diffracted from this crystal, it is not completely recorded on this detector molecule, because I already told about the X-ray have the very short wavelength. So to obtain the maximum resolution or the good separation between the diffraction spot, it is necessary that the distance between the crystal and the detector, it should be between the 150 nanometer. So This is according to Bragg's law. And the last step is the data measurement and their processing. This is the most important step, and generally it is carried out using various online suites, various online transforming softwares which is available, and this is one of the bioinformatics activity to obtain the atomic and the molecular 3D structure of the protein. So X-ray crystallographer is work up to the, it record the data on the detector in the form of spots, in the form of intensities of spot which is diffracted from the X-ray protein crystals. So here the from detector film, the data is generated, the electron density map is generated with the help of various transforming softwares, and that electromagnetic electron density maps get used to predict or to identify the molecular structure of the protein. Generally, HKN 2000 suite is used to process this raw data and to obtain the higher resolving 3D model of the protein. So this is all about the X-ray crystallography. In that we see what is X-ray crystallography, what is the need of X-ray crystallography, and what is the procedure and what is the mechanism of the X-ray crystallographer. Hope that you understand all of this, that, and it will be beneficial for your examination. Thank you. Thank you so much.